Hi, I'm Dongyoung Kim, an undergraduate student at our university. Here I'm going to present my ongoing study. The research topic is a comparative study between the cave and surface populations of Korean cryptic harvestmen using morphometrics and mitochondrial DNA. And now I'm going to start. The troglomorph is a common morphological characteristics found in cave-dwelling organisms, supposedly due to adaptive evolution. Caves vary widely throughout the world and often lack geographic connectivity among them, and the taxonomic range of cave-dwelling organisms is very large. Nevertheless, cave dwellers exhibit similar morphological traits because of the similar selective pressure of the special and common environment of caves, such as apodic conditions. Here are the fish, pseudoscorpion, and the coleoptera adapted to the cave environment. They are all depigmented and eyeless. The result of the convergent evolution, such as observed in these cave lives, is called troglomorphism. I think the key word of this study is troglomorphism, and what I focused on is the troglomorph observed in Opiliones. The Opiliones is a soil invertebrate commonly called harvestmen. They are a member of Arachnid. The class contains spiders, mites, ticks, scorpions, etc. Opiliones are often confused with spiders, but when you look at the spiders and harvestmen simultaneously, you can notice that the body of spider is divided into two parts, whereas the harvestman has one integrated body. And also, harvestman does not produce both poison and spider silk. The order Opiliones can be broadly classified into these four suborders, and in the suborder Laniatores, there are the family Paranichidae, which fascinated me. These microhabitat specialists favoring cool and moist conditions are the Beringian taxon found throughout Korea, Japan, the United States, and Alaska. Although some of them are geographically far apart, they show similar morphology due to their conservative ecological niche. They are small predators and sometimes scavengers found in caves and under the log and the litter. And the members of Paranichidae are known as dispersal-limited organisms having high regional endemism and low vagility. Among the several genera under Paranichidae, there is a Korean endemic genus Kaolinonychus. They are about 1 millimeter in size, found in most of mainland of South Korea except for the island, and their morphology looks similar to the other genera of Paranichidae. They are historically monotypic genus consisting of one species, Kaolinonychus coreanus, and two subspecies under it, Kaolinonychus coreanus and coreanus longipes. When I studied them with both my field experience and the description paper by Seishi Suzuki, I think it needs to be further supplemented. Although Suzuki's classification and description were truly insightful and also accurate, because there are few discussions about their environmental habitat and ecology. They are found both in caves and outside of cave surface, and these two types grouped by habitat show different morphology as my experience, but they are undescribed. That is, the current taxonomic system lacks its ecological trait. So I wondered how much these two environmental groups are different and what kind of relationship they have and how the troglomorphs have evolved in the cave populations. Following these questions, I thought that quantifying these morphological differences and identifying how these differences are associated with the genetic data can be the first step and provide fundamental information for further research related to my questions. For the study, we used 44 males sampled from 70 sites across South Korea. And the groups were divided based on whether the environmental habitat is cave or surface, regardless of the existing subspecies classification. Then we collected morphological and genetic data for each sample. The morphometrics data of 65 variables were generated by measuring the length measurements as much as I could. Meanwhile, mitochondrial C1 gene sequences were obtained for genetic data, and the matrix data were formed from SMP SNP subit. Since then, various multivariate analyses, including unsupervised machine learning and phylogenetic and population genetic analysis, were implemented using the obtained data. Now let's see the result from the panatic analysis. This is a plot of the t-SNP clustering using morphometric data. The figure on the left shows clustering with their absolute length data. You can see that there are 3 major clusters. What's interesting is that there are fusion populations between cave and surface populations, as well as clearly divided clusters of each groups. And this time we divided these length measurements into body lengths and converted them into proportional data, and then proceeded with clustering again. It is interesting that this linear distribution form is generated from the t-SNP method. And it is also inspiring that caves and surface populations are continuously divided. When I look at these results, I could deduce 2 things. First, cave and surface populations differ significantly in morphology. Second, that difference is continuous in proportional measurements. It doesn't seem like the 2 environmental groups have undergone a completely separate evolutionary history. Next, let's look at the PCA analysis result. The result of PCA, principal component analysis, also show that they are clearly divided. One thing to note is that compared to the cave population, the surface populations are clustered within a narrow range. Presumably, as I mentioned earlier, it is expected to have high morphological conservatism among various geographic populations due to the strong ecological constraints, even if not the cave population. Rather, in the case of cave population, the tendency of differences is coherent, but the degree of convergent evolution they have undergone in the cave environment is different, because the time of cave colonization and the period of adaptation is different for each other population. And this may cause relatively high morphological variances of cave populations. As I suppose, this is why morphological ranges are different between the cluster of the cave and surface population. Now let's go back to the PCA plot. You can see that these 2 environmental groups are distinctly separated by the x-axis, the principal component 1. Following it, we tried to track the loading values of what variables contributed to the principal component 1 and detect what characteristics distinguish the 2 environmental groups. These are the loading values of the principal component 1. We checked which variables were ranked high and conducted statistical tests from here. As a result, all the top 9 measurements showed statistically significant differences as shown. So let's take a look at what these variables are. When we look closely at these measurements, we were able to notice that they are commonly related to the length of appendages. Among them, the total length ratios of all 4 walking legs are included in the upper length. In other words, the cave population and surface population showed very clear differences in the length ratio of the walking leg. We could identify the significant differences between the cave and surface populations. If so, what are the characteristics with the least difference? What are conservative phenotypes between them? This time, let's look at the loading value linked below. As a result of statistical tests to them, there was no statistically significant difference in each measurement between the 2 groups. And also on the box plot, we could find that they were conservative. These characteristics included width of trochanter, the thickness and length of the short segment of the appendage, and the width of the body scutum, etc. And one interesting thing was that there was no difference between the height of the eye and the distance between the eye. In general, we think that the dark environment of the cave will give strong selection pressure to the visual ability, but we couldn't find any obvious evidence about it. And these measurements might have been inappropriate data to discuss this, though. In summary, between the 2 environmental groups, body width, length and thickness of some short segments of appendages, and eye-related length measurements were conservative. As a final step in the analysis of morphological data, I implemented partitioning around methods for classifying the sample in cluster optimally in a statistical way instead of labeling them with prior information. First, when we calculated the average slit width, we could find that it is optimal when the samples are divided into two clusters. Here is the result. Except for the 4 sample of cave population, symbol cave at the arrow, the result is very similar to those who were labeled according to environmental groups. So finally, it can be said that their morphologies are significantly distinct following the environmental habitat. And next is generic analysis. First of all, each phylogenetic tree of the mitochondrial CO1 gene. We estimated and compared 2 phylogenetic trees using 2 generally used algorithms, Bayesian inference and maximum likelihood. Now, what's the fact common in both phylogenetic trees is that the difference in environmental habitat appears to have occurred independently several times among the tree, not occurred by diverging within the branch of the tree. This is also shown by the dimensionality reduction methods, which is that the 2 environmental groups are not clearly distinguished, unlike the morphological data used in both PCA and t-SNE clustering. Especially in the PCA, the surface groups are somewhat clustered, but each cave population is distributed in its own location far apart from the others. So what criterion can they be grouped by? As the simplest idea, we divided each individual into 4 groups according to the clades identified in the phylogenetic tree. When we labeled each clade in a different group, we could see that the clusters are cohesive. Actually, it's a natural result because we use the same genetic input data, so similar results would be expected. And the same goes for t-SNE clustering. It's noteworthy that these clusters are not distinguished by environmental groups, whether they are caves or surface, but they are somewhat consistent geographically. If you look at the map on the right, you can see that the inner clade, yellow and green clade, is largely divided into the northern and southern. And also you can see the blue and red clade outside, which are inhabiting the caves that diverged earlier. This black-lined region is karst terrain, and it is estimated that these geographical factors may have caused their relatively early geographic isolation, as I suppose. One step further from here, I wanted to deduce how much gene flow exists between each environmental group and within each group. Due to incomplete lineage sorting phenomenon, estimating their gene flow with the CO1 gene alone produce low reliability information. But nevertheless, we conducted it for identifying some tendencies that could be further investigated in the future. The results are interesting because these 3 surface populations that you see here are geographically distant, but high levels of gene flow have been observed, while cave groups have identified nearly 0 gene flow with other nearby cave groups. Rather, gene flow between some caves and nearby surface populations was observed. This is considered a good example of the fact that the environment of caves actually causes high levels of geographic isolation. Meanwhile, it's also remarkable that there is a higher level of gene flow than expected in the surface population. But it does not say that surface populations are continuously connected. It is still difficult to tell these surface populations as a kind of panmixia state. I believe that more in-depth inference will be possible through more gene sequencing, and more thorough sampling. In conclusion, as suggested earlier, within the genus Kaolinonychus, the cave population and surface population show distinct morphological differences. And among them, it was also confirmed that the walking legs were clearly long in the cave group. It's presumed to be associated with cave adaptation related to sensitization in apotic conditions. On the other hand, when we did the cladistic analysis using the mitochondrial CO1 gene, their genetic clusters and lineages and clades were not highly related to environmental groups, but rather cohesive with geographic distance. But if you look at their gene flow, it doesn't seem that the type of environmental group has no effect at all. There are certain gene flows between surface groups, especially in some surface groups where relatively high gene flow has been observed, even at a distance. And on the other hand, it was estimated that there was little gene flow in cave groups. That's the discussion. One of the most important things we have seen in this study is the relationship between phenotype and genotype in cave adaptive evolution. Taken together, their morphological and genetic distributions tended to be incongruent. Based on this, we can also think about the relationship between genotype and phenotype in the evolution of cave adaptation. This can be inferred because cave adaptive evolution is, as is known, due to strong convergent evolution. It's not the xenomorphy they acquired when they underwent ecological isolation at some point, but the traits were acquired independently in different lineages. We estimated the relationship between cave adaptive phenotype and genotype in the subterranean harvestmen of the Korean Peninsula. However, there are still many questions that have not been answered. For example, we have seen in the research, environmental groups influence their gene flow, and their genetic profiles are related to geographic distance. Then how do identify and understand this complex evolution that takes into account both of these factors? This will be the most important question and direction of the future research. It seems important for further study to use more various gene or method. In particular, I hope that it will be suitable to use rod-seq, a widely used method to explore the history of genes at the population level. So looking forward to further advanced study, I will end this presentation. Thanks for watching.